All right, I know we already did this slide. It was the last slide from the last video, but I realized that I forgot to point out why photosystem 1 is receiving energy here in the form of light, photons, at the end of the electron transport chain. So the electron transport chain is running in between photosystem 2 and photosystem 1. So photosystem 2 is absorbing light energy, and that's exciting those electrons that run through the electron transport chain. But as they run through the chain, they're powering the pumping of protons, and that means they're losing energy. So by the time they get to photosystem 1, they're lower energy electrons, and the absorbance of light by photosystem 1 re-energizes those electrons, takes them to a higher energy state before they're picked up by NADP+ to form NADPH, so that those electrons carried by NADPH are high energy electrons. So that's what's going on at the end. So speaking of the light-dependent reactions, those are taking place here in the thylakoid membrane. So what we were just looking at on the previous slide is a molecular level view of what's going on in the thylakoid membrane. And those are the light-dependent reactions. So the light-dependent reactions take place in the thylakoids, and more specifically within the membranes of the thylakoids. But it also involves the space inside the thylakoids and the space outside the thylakoids, in other words, the stroma. But it starts with light energy being absorbed by chlorophyll in the thylakoid membranes by those photosystems. And what you need to concern yourself with when it comes to these processes that take place within the chloroplast is what goes in and what comes out of each process. So what goes into the light-dependent reactions obviously is light, but also from the environment comes water. And if you recall, at the very beginning here, water is split and oxygen is released. That's where the oxygen that plants release or breathe out, so to speak, comes from water. If you'll recall, the hydrogen ions and the electrons from water are what the plant actually wants to get during the light-dependent reactions. Those are the electrons that are flowing through the thylakoid membrane, pumping hydrogen ions and allowing for chemiosmosis and the photophosphorylation to create ATP. Speaking of which, ATP then is a product along with NADPH, which is carrying high-energy electrons as well as hydrogen. But there are also hydrogen ions that are available, that were made available from the splitting of water. So what comes out? Oxygen, just as a byproduct, ATP, which is chemical energy, NADPH, which is chemical energy in the form of electrons, hydrogen ions, that all goes to the stroma, what surrounds the thylakoid membrane, the space that surrounds the thylakoid membrane. So all of this, the whole chloroplast, is filled with aqueous solution that we call the stroma. But of course there are enzymes in that stroma that carry out what's known as the Calvin cycle, or the light-independent reactions. And in the light-independent reactions, that's where the energy, the chemical energy coming from the light-dependent reactions, is used to fix carbon, this all-important ecological activity that fixes carbon for the entire biosphere on the surface of the Earth. So carbon dioxide, what that refers to, is taking carbon from carbon dioxide and incorporating it into organic compounds, eventually like glucose. So that's carbon fixation. So carbon fixation, when carbon dioxide, which is considered inorganic carbon, is taken in during the Calvin cycle or the light-independent reactions, it's incorporated into organic compounds, and that's what's referred to as carbon fixation. There's an enzyme that carries this process out, or performs this process. It's known as Rubisco. And there are a lot of enzymes that are functioning in the Calvin cycle, but that's the only one that I expect you to know. It is the carbon-fixing enzyme that's functioning in photosynthetic organisms that allows them to take in carbon dioxide and incorporate it into organic compounds. So it's an extremely important enzyme on the planet because it's responsible for ultimately the production of food for everybody. And hopefully you recognize this by now as glucose. In the meantime, ATP has given up its energy, NADPH has given up its energy, and so those are going to be recycled as ADP and NADP plus back to the light-dependent reactions to be re-energized, to be recharged back into ATP and NADPH so that they can deliver that energy to the Calvin cycle. And the whole thing just continues on with an ATP-ADP cycle and an NADPH-NADP plus cycle going on between the light-dependent and light-independent reactions. So here's everything that I just explained, basically. I'm not going to read through it for you. You can read through it yourself. What's on the next slide is a more detailed look at what's really going on during the Calvin cycle. All right, I know this looks kind of daunting, and actually, believe it or not, it can get a lot more detailed than this and complicated than this. So we're going to try to simplify this as much as possible. But this shows the Calvin cycle, and what I want you to note right off the bat here is just ignore these numbers out front. The 6 and 12s, and 10s, just ignore those. They're just going to confuse you. So hopefully I'll be able to explain this and not confuse you too much. So as I pointed out on the previous slide, the Calvin cycle operates just like all biochemical pathways. You know, it's a biochemical pathway, and for every reaction that takes place in the Calvin cycle, there's an enzyme that catalyzes that reaction. So enzymes run the whole thing. And remember, enzymes are proteins, and so I've told you many times, proteins run the show, and enzymes are pretty much how proteins run the show. So the most important thing that's going on here is taking carbon dioxide and incorporating that, adding it to an organic molecule, and this is the organic molecule that carbon dioxide is added to. It's called rubulose bisphosphate, and you don't need to know that, but that process of adding carbon dioxide to rubulose bisphosphate at the end, which is the last molecule at the end of the cycle here, is what we refer to as carbon fixation. That's incorporating carbon dioxide into that organic compound. Rubisco is the enzyme that performs that. So that's rubulose bisphosphate carboxylase. Rubulose bisphosphate carboxylase, but yeah, so you can see why they call it Rubisco for short. And really what Rubisco is doing, well, in the diagram here, you'll notice that we're going from we're adding a carbon dioxide onto a 5-carbon compound. So we're adding 1 carbon onto this 5-carbon compound. So that should form a 6-carbon compound, but that's not being shown in this figure. It's not being shown in this diagram. So what's really going on is that Rubisco is adding that carbon to rubulose bisphosphate, creating this 6-carbon compound, and that immediately splits into 3-carbon compounds. So that's how we get what we're looking at here. The Rubisco forms this 6-carbon compound, which immediately splits into 3-carbon compounds. And that's where we are right here. Next is where further magic happens, and you'll notice that ATP is involved here in NADPH, and if you'll recall, those are coming from the light-dependent reactions. Basically, this is the energy of light. The light energy has been converted to ATP and electrons in NADPH, so those are from the LDRs, the light-dependent reactions. And if you'll recall, the way the ATP does its magic is through phosphorylation. So these ATPs, the ATP that comes in here is phosphorylating this 3-carbon compound, and that energizes it. Don't forget that there are hydrogen ions from water that are coming from the light-dependent reactions as well. So, and along with NADPH, which is what we call reducing power. It's going to add electrons here. So that's energy. That's adding energy. So both ATP and NADPH in this step of the cycle are energizing this 3-carbon compound. So we go from a 3-carbon compound that doesn't have a whole lot of energy to a 3-carbon compound that is relatively highly energized, and that's known as G3P, glyceraldehyde 3-phosphate. And you'll know, because it's a cycle, it goes round and round and round. So we're going to come back to what comes out of the cycle in a minute. First, we're going to take a look at how the cycle continues. And the cycle continues as most of the G3P continues on in the cycle. And so that's the same G3P that we have here. And then it gets energized further by ATP. And where does the ATP come from? The light-dependent reactions. So you can see the importance of capturing light energy in the light-dependent reactions to make ATP, because a lot of ATP is used in the process, in the Calvin cycle, to create carbohydrates. So the ATP energizes the G3P, and I'm not going to go into the details, but eventually that creates these 5 carbon compounds through phosphorylation and enzymes, of course, creates the 5 carbon compound that we started out with, ribulose bisphosphate. So this part of the cycle is referred to as regeneration, because the whole reason for using ATP here is to regenerate the compound that's necessary for Rubisco to be able to add a carbon to and continue the cycle. So this compound, ribulose bisphosphate, is really important to the cycle, because without it, ribulose bisphosphate carboxylase won't have anything to add carbon to. And that completes the cycle. Now we can come back to what's going on as far as the product. So again, most of the G3Ps continue on, but some G3Ps exit the cycle. And it takes 2 G3Ps, because G3P, glyceraldehyde 3-phosphate, is a 3-carbon compound. It takes 2 of those to make 1 molecule of glucose. And of course, enzymes are responsible for performing this. And it's not only glucose that can be created here. G3P is like a building block, a carbohydrate building block, and it can be combined in various ways to create all kinds of different carbohydrates. So it says here sugars like glucose and other compounds, other organic compounds, other carbohydrates, but not only carbohydrates, other organic compounds also. It all depends on the enzymes and the biochemical pathways that are using G3P as a building block as to what the product's going to be. So the bottom line is that plants are biochemists. They're able to create organic compounds. They're able to form all kinds of different organic compounds with G3P as a starting material, and that all, if you'll recall, began with light energy. Light energy is what enables them to make the G3P, and then they have all these enzymes that are able to create all kinds of compounds that we all need, all us living things on the planet, on the surface of the planet anyway. All right, so everything that I just explained is right here. 5-carbon compound regenerated from the cycle. Rubisco fixes CO2 to the 5-carbon compound regenerated from the cycle. The 6-carbon compound, which is here, splits into 2 3-carbon compounds. That's converted and energized by ATP and NADPH to G3P, right? So now we have G3P. The biochemical building block product of the Krebs cycle, or I'm sorry, the Calvin cycle. The light independent reactions. And most G3P cycles goes on and it gets regenerated back into ribulose bisphosphate, and ATP is required for that, and then the cycle starts over again. And the G3P product, of course, is used to make a variety of organic compounds, like glucose. The last thing here is a final note, just some of the big words that I use, like ribulose bisphosphate and ribulose bisphosphate carboxylase and glyceraldehyde 3-phosphate. So those are all written down here so you know how they're spelled. But what you need to remember is Rubisco and G3P, and of course glucose, C6H12O6. The acronyms make it a lot easier. The next slide is a summary of the entire process. So we have the thylakoid membrane here, we've got the stroma out here. So we're seeing both the light-dependent and the light-independent reactions and how they're linked together by the ATP-ADP cycle and the NADPH-NADP plus cycle. So, as I'm suggesting here, you refer to the diagram to write a summary of photosynthesis, the entire photosynthetic process, both the light-dependent and light-independent reactions. That would be a great exercise for you to perform as a way to practice and solidify what you understand about photosynthesis.